Nutrition. Nutrition is the overall process by which living organisms synthesize or procure their food, utilize it for maintenance and work, and convert it into protoplasmic constituents for growth, health, repair, regeneration, and reproduction. So, nutrition is the overall process by which living organisms synthesize or procure their food, utilize it for maintenance and work, and convert it into protoplasmic constituents for growth, health, repair, regeneration, and reproduction. It involves a series of biochemical and physiological changes by which the components of food are converted to the constituents of the body. The energy giving and body building constituents of food are called nutrients. Functionally, nutrients fall under three groups, namely energy suppliers, bodybuilders, and regulators. Energy suppliers include carbohydrates and lipids. Bodybuilders include proteins, water, and minerals. Regulators include vitamins and some minerals. So, energy suppliers, bodybuilders, and regulators. Energy suppliers include carbohydrates and lipids. Bodybuilders include proteins, water, and minerals. Regulators include vitamins and some minerals. Bodybuilders and energy suppliers are needed in relatively large amounts, and so they are often called macronutrients. On the other hand, regulators are required in small amounts, and so they are called micronutrients. Organisms of different evolutionary descent and taxonomic stages have almost the same elemental requirement. They all require the same elements for maintenance, repair, and growth. However, the mode of nutrition is different in different groups. This difference is primarily due to the diversity of raw materials from which the elements are obtained, and also due to the variability of the methods employed for obtaining the elements. Next topic, classification of nutrition based on the source of nutrients. Based on the source of nutrients, two major kinds of nutrition can be recognized, autotrophy and heterotrophy. In the former, organisms synthesize organic nutrients all by themselves, whereas in the latter, they get them from other organisms. So, autotrophy or self nutrition is a nutrition in which organisms synthesize organic nutrients from inorganic constituents such as water, minerals, CO2, NH2, nitrates, etc. So, it is a type of nutrition in which organisms feed themselves independent of others. Autotrophs include some bacteria, some protists, algae, and all green plants. For survival, autotrophs require only water, CO2, inorganic salts, some minerals, and a source of energy. The source of energy is different in different autotrophs. Based on the source of energy used for the synthesis of organic nutrients, two types of autotrophy can be recognized, namely chemotrophy or chemoautotrophy or chemosynthetic autotrophy, and phototrophy or photoautotrophy or photosynthetic autotrophy. First, chemotrophy. Chemotrophy is a autotrophy in which organisms synthesize organic nutrients from inorganic constituents by chemosynthesis, utilizing the chemical energy obtained by the oxidation of electron donating inorganic or organic substances such as H2S, NH2, nitrates, iron, sulfur, etc. The chemotrophs, which obtain energy by the oxidation of inorganic molecules, are called lithotrophs, and those which obtain energy by the oxidation of organic molecules are called organotrophs. Chemotrophs mostly live in hostile environments or tough situations such as hot springs or geysers, deep ocean vents, salt marshes, etc. So, chemosynthetic autotrophs or chemoautotrophs include some bacteria such as methanogenic bacteria, halophilic bacteria, thermoacidophilic bacteria, nitrifying bacteria, iron bacteria, and sulfur bacteria. These bacteria have special enzymes which can catalyze the oxidation of substances and can also couple each reaction with the generation of energy rich phosphates. This nitrate bacteria can oxidize nutrients to nitrates. Iron bacteria can oxidize ferrous ion to ferric ion. Sulfur bacteria can oxidize H2S to sulfates. The energy derived from these oxidations will be utilized for synthesizing organic substances from CO2, H2, and nitrogenous compounds which they have already absorbed from the surrounding medium. The energy derived from the oxidations will be utilized for synthesizing organic substances from CO2, H2, and nitrogenous compounds, which they have already absorbed from the surrounding medium. Then phototrophy. Phototrophy is the autotrophy in which organisms synthesize organic nutrients from inorganic constituents by photosynthesis with the help of solar or radiant energy and chlorophyll. It involves the conversion of radiant energy to chemical energy and its conversion in the chemical bonds of organic molecules. So phototrophs or photosynthetic autotrophs or photoautotrophs include chlorophyll-bearing organisms such as green plants, green algae, brown algae, some red algae, diatoms, dinoflagellates, cyanobacteria, green bacteria, purple sulfur bacteria, purple non-sulfur bacteria, etc. Bacterial photosynthesis differs from that of the phototrophs in two major aspects. It occurs by absorbing light from the near infrared portion of the light spectrum rather than from the visible spectrum. It is not oxygenic since water is not used as a hydrogen donor. Sulfur bacteria utilize H2S as the hydrogen donor. Then heterotrophy. Heterotrophy is the nutrition in which organisms procure performed organic nutrients from other living or dead organisms or their products. Heterotrophs are unable to synthesize organic nutrients from inorganic constituents using free energy, and that is why they entirely depend upon the organic nutrients from other organisms. All animals and fungi, most protists, and some bacteria are heterotrophs. All heterotrophs often energy-rich nutrients utilize and ultimately from phototrophs, which can trap radiant energy to synthesize organic compounds. Next, let's learn about classification of heterotrophy based on the source of nitrogen requirement. Heterotrophic nutrition is classified variously based on different aspects based on the source of nitrogen requirement. Two kinds of heterotrophic nutrition can be recognized: mesotrophy and metatrophy. In both these, amino acids are the source of nitrogen. Autotrophs make use of inorganic nitrogen, nitrates, or ammonia as the source of their nitrogen requirement. In mesotrophy, a single kind of amino acid serves as a source of nitrogen requirement, whereas in metatrophy, the source of nitrogen requirement consists of several kinds of amino acids, either as compounds or as mixtures. Mesotrophs include some protists and metamorphs include animals. Classification of heterotrophy based on the nature of the food. Based on the nature of the food and the mode of feeding, 5 kinds of heterotrophy can be recognized, namely mutualism or symbiotic nutrition, parasitism, saprotrophy, osmotrophy, and holotrophy. The condition in which an organism resorts to more than one of these methods is called mixotrophy. So mutualism or symbiosis. Mutualism or symbiosis is a mutually beneficial nutritional reaction or a relation. Mutualism or symbiosis is a mutually beneficial nutritional relation between two different species. It is of two kinds, namely obligatory mutualism and facultative mutualism. Obligatory mutualism is a compulsory relation in which neither species can survive free from the other under normal natural conditions. The association between termites and the protozoan Trichonympha campanula is a classical example. Trichonympha inhabits the alimentary canal of termites, deriving nutrients and other biological requirements. In return, it benefits termites by digesting the cellulose content of their food with the help of its enzymes. Facultative mutualism, also called protocooperation, is a voluntary association in which one or both of the species can live without the other. The association between some birds and grazing mammals is an example. The birds feed upon the ticks, which are ectoparasites on cattle. The association between man and some intestinal bacteria like Escherichia coli is particular in that it is obligatory for bacteria but facultative for man. These bacteria inhabit the colon of man. Man provides them simple food. In return, the bacteria synthesize some vitamins for human. They cannot live outside human intestine, but man can live without them. So parasitism or parasitic nutrition is the nutrition in which an organism or parasite feeds upon the food, body fluids, or tissue of other host without necessarily killing it. It can be obligatory or facultative and temporary or permanent. Parasitism is ecologically significant in that it plays an important role in maintaining the balance of nature, and also in that it promotes adaptive specializations in the parasites for a successful parasitic life. Then saprotrophy. Saprotrophy is the absorption of simple inorganic solutions or solutions of dead and decayed organic matter through the body surface. Saprobes include some bacteria, some fungi, some plants, and a few protozoans. They can grow only in place where there are decomposing parts of animals or plants, or masses of plant and animal byproducts. Saprobes may secrete digestive enzymes to the outside to hydrolyze complex organic matter to simple soluble and absorbable products. Then osmotrophy. This is the nutrition in which pre-digested food is absorbed through the body surface. This is found in parasitic forms such as trypanosoma and Taenia. Pre-digested food is absorbed through the body surface. Osmotrophy. A new term, osmotrophy. Pre-digested food is absorbed through the skin or the body surface. For example, in case of trypanosoma and Taenia. Then holotrophy. Holotrophy or holozoic nutrition is the nutrition in which complex solid or liquid food materials are taken into body and digested inside. This is common among animals and most species. Protists, organisms exhibiting holotrophy are known as holotrophs. So let's classify holotrophy. Based on food and feeding habits, holotrophic nutrition is variously classified. Some common types of holotrophy are following, like herbivory. Herbivory is a habit of feeding on algae or plants. Cattle, sheep, goat, camel, deer, zebra, giraffe, horse, rabbit, monkeys, apes, etc. are herbivorous mammals. Herbivores may restrict their diet to a particular plant part such as leaves, fruits, seeds, roots, nectar, etc. Similarly, a herbivore may feed either on one or on several species of plants. Many aquatic herbivores are ciliary and mucoid filter feeders. They create an incoming current of food-laden water by sea derivating and trap from it tiny food particles with the help of mucus secretion. Then carnivory. Carnivory or predation is the habit of feeding on other animals or on their flesh and bone. A number of invertebrates and many vertebrates are carnivores. Carnivores are predators specialized for locating and capturing their prey. Even simple carnivorous invertebrates are provided with such adaptations. For example, coeluroids have tentacles and nidoblasts for catching and paralyzing the prey. Carnivorous vertebrates also have many adaptations for catching the prey. The shooting tongue of frogs and some lizards, the sharp teeth and claws of lions and tigers, etc. Specialization to catch and kill the prey. Apart from this, carnivorous mammals have highly specialized canines for tearing flesh, and their molars are specialized for shredding meat into small chunks. The alimentary canal of carnivores is relatively shorter. The reason is that the food needs not be retained for a long time in the digestive tract, since meat can be more easily digested than plant food. Then next, omnivory. Omnivory is the nutrition in which the food consists of plant and animal materials. Cockroaches, crows, bears, human beings, etc. are omnivorous. Omnivores are generally adapted to discriminate between different smells and tastes. This adaptation enables them to select their food. Then insectivory. Insectivory is the habit of feeding on insects. Frogs, toads, lizards, some bats, etc. are insectivorous. Sanctuary or hematophagy is the blood feeding habit. Leeches, bedbugs, mosquitoes, some flies, parasitic ticks, mites, and fleas, vampire bats, etc. are sanctuaries. The salivary enzymes of sanguivores can prevent blood clotting because mostly that contain hirudin in case of leeches. Then frugivore. Frugivore is the fruit eating habit. Some birds, some bats, monkeys, etc. are frugivores. Then granivore is a grain feeding habit. Pigeons, parrots, doves, sparrows, etc. are granivores. Then graminivore in the case of feeding on grasses. Cattle, goat, grasshoppers, etc. are grass eaters. Then detritivores is a habit of feeding on detritus, decaying organic matter. Earthworms are detritivores. And then myrmecophagy. Myrmecophagy is the habit of feeding on ants and termites. Spiny anteater or echidna, scaly anteater, manus, greater anteater, Myrmecophaga, lesser anteater, tamandua are myrmecophagous mammals. Anteaters in general have two specializations, namely large, powerful, and strongly curved claws of the front legs for digging and ground and also for breaking ant hills and termite area. Enormously elongated snout and long, thin, protrusible, and sticky tongue for collecting ants and termites. Then coprophagy. Coprophagy is feeding, also known as refaction or pseudo rumination, as a special mode of feeding, evolved as an adaptation for cellulose digestion. It is found in rabbits, hares, guinea pigs, and many rodents. In this animal, cellulose digestion takes place in the cecum with the help of enzymes secreted by certain symbiotic bacteria. Cecum is beyond the region of actual digestion and absorption. In coprophagy, freshly eaten food straight away passes to the cecum without staying in stomach and intestine. In the cecum, cellulose gets digested by bacterial enzymes. The partly digested contents of the cecum form a sort of soft feces. It will not be dropped by the animal but will be eaten directly from the anus. This re-injection enables a complete digestion and utilization of the food. The soft feces food will not be masticated, and in the stomach it will not be mixed with other food. It will remain in the fundus part of the stomach for many hours for complete gastric digestion. This fundus serves as a digestive center analogous to ruminants. From the stomach, the re-ingested food slowly passes to the intestine where final digestion and absorption occur. This time it will not re-enter the cecum but will pass to the rectum and drop out as fecal pellets. Coprophagy is significant in that it enables a more complete digestion, absorption, and utilization of the food. Also, it provides a rich source of vitamin K and biotin and increase the growth rate of the animal. Re-ingestion of feces has been reported from some shrews, marsupials, and prosimians also. Then fluid feeding. This is the habit of feeding on plant sap and nectar. Honeybees, butterflies, and several other insects are fluid feeders. Their mouth parts may be of piercing and sucking type. Then stages of holotrophy. Holotrophy involves 6 successive processes, namely food capture, ingestion, digestion, absorption, assimilation, and digestion. Ingestion is the taking in of food materials into the body. Ingestion of microscopic food is called microphagy, and that of large food is called macrophagy. Within the body, the food is converted into an absorbable form. This is called digestion. The digested food constituents are soon taken into the body fluid. This is absorption. Some of the absorbed food constituents would be utilized for the synthesis of bodybuilding protoplasmic constituents. This is assimilation. The remainder would be utilized for an energy supply. A small share may be stored within the body as reserve food. The undigested residue is called feces. It is passed on to the outside of mechanical means. This is called digestion or defecation. Next, changes of food in the alimentary canal. Now we are trying to learn about digestive system. So changes of food in the alimentary canal. In the alimentary canal of macrophagous feeders, complex food undergoes three stages of changes: mechanical, chemical, and physical. Mechanical changes bring about the reduction of complex and massive food into small pieces. Chemical changes bring about the digestion of the reduced pieces into absorbable constituents. Physical changes involve the absorption of the end product of digestion into the body fluids. Digestion is the breaking down of complex, unabsorbable, unassimilable, and unmetabolizable organic macromolecules or macronutrients into simple, absorbable, assimilable, assimilable and metabolizable micromolecules or micronutrients. The micronutrients undergo going digestion include complex carbohydrates, proteins, lipids, and nucleic acids. The products of their digestion are absorbed or assimilation for assimilation and oxidation. Digestion is characteristic of most heterotrophs with the exception of saprotrophs and osmotrophs. So intracellular and extracellular digestion. Digestion is of two main kinds: intracellular and extracellular or intercellular. Intracellular digestion occurs inside the cells within food vacuoles, so it is also termed vacuolar digestion. In it, the food vacuoles containing ingested food fuse with lysosomes. Soon their contents are digested by lysosomal hydrolysis. Digestion is entirely intracellular in protists and sponges, but in coelenterites and ctenophorans it is partially intracellular and partly extracellular. In this case, the initial phase of digestion is extracellular, taking place in the coelenteron. The products of this preliminary digestion are soon engulfed by some amoeboid phagocytic endodermal cells. The final phase of the digestion takes place in the food vacuoles of these cells. In some higher invertebrates such as platyhelminthes, nemerteans, annelids, mollusks, and echinoderms, intracellular digestion supplements extracellular digestion. In most of them, intracellular digestion is carried out by certain enzyme-filled wandering amoebocytes found in the gut wall. Intracellular digestion is rare or absent in nematodes, insects, crustaceans, and cephalopods. Among chordates, intracellular digestion occurs occasionally in Amphioxus. Extracellular digestion occurs outside the cells, either in a digestive cavity or in the lumen of the alimentary canal. In general, it is completed in three stages, namely conduction and storage of the ingested food, initial digestion, and final digestion and absorption. So extracellular digestion has advantages in that it permits the digestion of large pieces of food. Intracellular digestion is reversed only for small particles which can be taken into individual cells. So elaborate extracellular digestion requires a well-developed digestive cavity or digestive tract for the action of enzymes on food. In general, it is completed in three stages, namely conduction and storage of the ingested food, initial digestion, and final digestion and absorption. So extracellular digestion has advantages in that it permits the ingestion of large piece of food. Extracellular digestion has advantages in that it permits the digestion of large piece of food. So intracellular digestion is reserved only for small particles which can be taken into individual cells. Elaborate extracellular digestion require a well-developed digestive cavity or digestive tract for the action of enzymes on food. In some cases, such as coelenterites, flatworms, and brittle stars, the digestive tract has only a single opening, the mouth. In such animals, both ingestion and digestion occur through the same opening. In more complex animals, there are two openings, namely mouth and anus, at opposite ends. In them, ingestion of the food occurs through the mouth and digestion of the feces occurs through anus. So the passage of food along the digestive tract will be continuous and uninterrupted. Then mechanism of digestion in macrophages feeders, digestion involves two sets of processes, mechanical and chemical or physiological. The mechanical part of digestion involves the reduction of large and massive solid food into small pieces by physical processes such as cutting crushing, tearing and processing, pressing and grinding, etc. It renders the physical process easy and effective. The mandibles of insects, gastric mill, or the internal masticatory apparatus of crustaceans, radula of mollusks, teeth of mammals, the powerful gut musculature, etc., are concerned with the mechanical reduction of the food. The mechanical part of digestion in most mammals include five major events, namely mastication, insalivation, deglutination, churning, and the backward movement of the food mass along the intestine. Mastication or chewing is the breaking down of the solid food into small particles by cutting, tearing, and grinding with the help of teeth and tongue. Insalivation is the mixing of the food with saliva. Mastication, insalivation, deglutination, churning, and the backward movement of the food mass along the intestine. So mastication and insalivation take place in the oral cavity. Deglutination is the swallowing of the food. Churning is the pressing and rolling of the food mass in the stomach cavity by mixing waves of the stomach wall produced by powerful peristaltic contractions. Churning brings about the thorough mixing of the food with gastric juice. The chemical part of digestion involves the enzymatic hydrolysis of large and complex food into small and simple constituents. This involves the uptake of water and the action of specific enzymes. Digestive enzymes fall under four major groups, namely carbohydratases or amylases, amylolytic or carbohydrate splitting enzymes, protease or proteolytic or protein splitting enzymes, and lipases, lipolytic or lipid splitting enzymes, and nucleases, nucleic acid splitting enzymes. So different types of digestive enzymes are involved, like carbohydrates, starch splitting enzymes, amylase, alpha amylase cleaves the alpha 1,4 glycosidic bonds, beta amylase remove maltose units, glycogen splitting enzymes, amylase, gamma amylase removes glucose units from glycogen, cellulose digesting enzyme, cellulase split cellulose to cellobiose. The secretion of this enzyme very seldom occurs in animals. Then chitin splitting enzymes, chitinase removes glucose units from chitin, oligosaccharide splitting enzymes, cellobiose splits cellulose to glucose units. The secretion of this enzyme very seldom occurs in animals. Then maltose splits maltose to glucose units. Isomaltase splits isomaltase to glucose units. Then lactase splits lactose to glucose and galactose. Then sucrase or invertase splits sucrose to glucose and fructose. Then protease endopeptidases hydrolyze the interior peptide bonds near the C terminus and thereby break down polypeptides into peptide fragments, pepsin, renin, trypsin, chymotrypsin. Then exopeptidases hydrolyze the terminal peptide bond and thus break down polypeptides to amino acid units like carboxypeptidases, aminopeptidases, dipeptidases. Then lipases hydrolyze the esters of long chain fatty acids, forming mono or diglycerides and fatty acids. Then esterases hydrolyze the esters of short chain fatty acids to form fatty acids and glycerol. Then nucleases, ribonuclease hydrolyzes RNA, forming ribonucleotides. Deoxyribonuclease hydrolyzes DNA, forming deoxyribonucleotides. Next, digestion of carbohydrates. Dietary carbohydrates are of two major groups, namely structural polysaccharides, which cannot be digested by the enzymes of vertebrates, and universally digestible polysaccharides. The first group includes cellulose, lignin or structural polysaccharides, lignin, cellulose, chitin, inulin, agar, dextrins, mannans, and pentosans, pectic acid, alginic acid, etc. The second group include starch and glycogen. Starch is a storage product of plants composed of amylose and amylopectin. Glycogen is the storage product of animals composed of glucose units. Carbohydrate digestion in vertebrates. In vertebrates, carbohydrate digestion occurs mostly in the small intestine by the action of pancreatic and intestinal carbohydrates. In some cases, it begins in the oral cavity also by the secretion and action of salivary enzymes. In such cases, the enzymes salivary amylase and salivary maltase initiate carbohydrate digestion. Salivary amylase hydrolyze starch to dextrins, isomaltose and maltose. So maltase converts maltose to glucose. Digestion of carbohydrate constituents continues further in the small intestine. In the small intestine, pancreatic alpha and beta amylase, amylopsins, hydrolyze digestible polysaccharides yielding disaccharides such as maltose, isomaltose, lactose, and sucrose. These are further acted upon by the intestinal disaccharide maltase, isomaltase, lactase, and sucrase or invertase. So maltase splits maltose to glucose units, isomaltase splits isomaltose to glucose units, lactase hydrolyzes lactose to glucose and galactose. Sucrase breaks down sucrose to glucose and fructose. Another intestinal enzyme widespread from fishes to man but absent in birds, cats, koalas, and pinnipedians, seals, sea lions, and polyneurises is trehalase. It acts upon the disaccharide trehalose, which is abundant in algae, mushrooms, and insects. For the intestinal disaccharides, lactase is absent in non-mammalian vertebrates whose young ones are not suckled. It occurs only in those mammals which secrete milk or synthesis lactose. Some marine mammals, notably pinnipedians, do not secrete lactose or any other carbohydrate in their milk, so intestinal lactase is absent in their young ones. Sucrase also is absent in pinnipedians and it's very low in certain fish eating birds. In mammals, with the exception of man, intestinal sucrase and maltase are produced only after weaning. In a number of ruminants which metabolize sugars in their intestinal lumen, these enzymes are low or absent at all stages. So oral digestion, starch, with help of salivary amylase, dextrins, isomaltose, and maltose are produced. Then maltose once again undergoes this process of digestion like salivary amylase, then transformed to glucose. Starch, salivary amylase, dextrins, isomaltose, and maltose, salivary amylase, glucose. Then intestinal digestion, polysaccharides, pancreatic amylase, maltose, isomaltose, lactose, sucrose, maltose, and intestinal maltase to glucose. Isomaltose, intestinal isomaltase create glucose. Then lactose, intestinal lactase, glucose and galactose. Sucrose, intestinal sucrase, glucose and fructose. Digestion of cellulose. Cellulose is a glucose polymer and most important structural element or material of plant cells. It is a water insoluble and highly refractory of chemical attack. Cellulose digesting enzymes are altogether absent in the digestive system of vertebrates, yet many of them digest cellulose and depend on it as their main energy source. Two cellulose digesting enzymes have been reported from the intestine of several invertebrates which feed on wood and plant products. However, in all vertebrates and most invertebrates, cellulose digestion is carried by certain symbiotic microorganisms inhabiting their digestive tract. These organisms are believed to produce two enzymes, cellulase and cellobiase. Cellulase breaks down cellulose to cellobiose, and cellobiase hydrolyzes cellobiose to glucose units. Symbiotic cellulose digestion has been reported from snails, termites, ruminant mammals, non-ruminant herbivorous mammals, etc. Shipworm or teredo and silverfish Tenolipisma are believed to digest cellulose by their own enzyme. Symbiotic cellulose digestion in snails and non-ruminant mammals is carried by the enzyme secreted by symbiotic bacteria. In ruminants, by the enzymes of symbiotic bacteria and protozoans, and in termites, by the enzyme secreted by the symbiotic flagellate protozoan Trichonympha. In ruminant mammals, rumen of the stomach is the center of cellulose digestion. In sloths and langur monkeys, stomach is multi-component and it serves as a center of cellulose digestion. In some marsupials and quarks, stomach is rumen-like for cellulose digestion. In other non-ruminant mammals, symbiotic cellular digestion occurs in the cecum. Then digestion of carbohydrates in invertebrates. Studies on the carbohydrate stasis of invertebrates is only incomplete and fragmentary, although amylases and disaccharides are widely distributed. In crustaceans and insects, the midgut gland widely secretes alpha amylase. In certain groups of insects, salivary alpha amylase also is secreted. Lactase is absent in most invertebrates, excepting some oligochelates, some snails, and a few insects such as honeybee larvae, cockroaches, locusts, and beetles. Also, the widespread occurrence of lactase among crustaceans has also been established. Sucrase activity is of wide occurrence among insects, especially in nectar feeding forms. The larvae of moths and butterflies have sucrase in addition to lipase and protease in the midgut, but their nectar feeding adults have only sucrase and no other digestive enzymes. Non feeding moths have no digestive enzymes at all. Carbohydrate digestion is usually weak in sanguivorous and carnivorous invertebrates. For example, some blood sucking flies, tsetse fly, have no salivary carbohydrates but have amylase and sucrase in the posterior midgut. On the other hand, some non-blood sucking flies have amylase in the salivary, amylase, maltase, and sucrase in the entire midgut. Herbivorous gastropods, which feed on phytoplankton, have high amylase secretion in crystalline style and hepatopancreas. On the other hand, in carnivorous gastropods, crystalline style is absent and there is only weak amylase production in hepatopancreas. Trihalase is present in the gut of many insects and some other invertebrates which feed on fungi, earthworms, and some other oligosaccharides whose diet include fungal spores. Next, digestion of proteins. Proteins are polymers of amino acid residues bound together by peptide bonds. We know that. The digestive enzyme which attack these peptide bonds are called proteases. Digestion of proteins follows a more or less uniform pattern throughout the animal kingdom. Initially, intact polypeptide units are broken down to smaller fragments by peptide-peptide hydrolases. Peptide-peptide hydrolases, formerly called proteinases. These enzymes attack the interior peptide bonds at various points of the peptide chain and never the terminal peptide bond. In the next stage, these peptide fragments are broken down to amino acids in a step-by-step manner by which set of enzymes which attack only the terminal peptide bonds. These protein digestive enzymes are of two major groups, namely endopeptidases, which attack the interior peptide bonds, and exopeptidases, which attack the terminal peptide bonds. So, protein digestion in vertebrates. Digestion of proteins in vertebrates is completed in three major phases. In the first phase, complex protein molecules or polypeptide units are broken down to small fragments or oligopeptides by the addition of endopeptidases. In the second phase, these fragments are broken down to dipeptidases in the step-by-step manner by a set of exopeptidases. In the final phase, these tripeptides and dipeptides are hydrolyzed to amino acids by a set of exopeptidases. The commonest endopeptidases found in vertebrates are pepsin, renin, trypsin, chymotrypsin, and elastase. The exopeptidases include carboxypeptidase, aminopeptidases, dipeptidases, and tripeptidases. So, aminopeptidases, dipeptidases, and tripeptidases are collectively called rupsins. Digestive protease, endopeptidase, exopeptidase, pepsin, renin, trypsin, chymotrypsin, elastase, and carboxypeptidase, aminopeptidase, dipeptidases, and tripeptidase. So, pepsin is a proteolytic enzyme which acts on positively charged proteins in an acidic medium. It may get inactivated in a neutral or alkaline medium. In vertebrates, pepsin is secreted by the gastric glands as an inactive precursor or proenzyme called pepsinogen. It is activated by the HCl of the gastric juice. Sometimes it may be activated by active pepsin also. This may be called the autocatalytic activation. Pepsin specifically hydrolyzes only those hydrogen bonds that are located in between the phenyl group carrying amino acid like tyrosine, phenylalanine, and decarboxylic acid, aspartic acid, glutamic acid. The action of pepsin results in the breakdown of proteins or polypeptides into several short fragments. Then renin is a milk coagulating enzyme found in the gastric juice of calves but absent in adults. It coagulates or curdles milk by converting the milk protein casein to paracasein in the presence of Ca2 plus ions. Paracasein precipitates as calcium procaseinate, which forms the curd. Then trypsin is the endopeptidase found in the pancreatic juice of vertebrates. It is most active in a slightly alkaline medium. It hydrolyzes the peptide bonds that are adjacent to a basic amino acid. The action of trypsin also results in the formation of short-chain oligopeptides. Pancreas secretes trypsin as an inactive prozyme called trypsinogen. Trypsinogen is activated usually by the active intestinal enzyme enterokinase. Often it may be activated by the active trypsin also. This is called autocatalytic activation. Autocatalytic activation. Okay. Then chymotrypsin. Chymotrypsin is a pancreatic endopeptidase, very much similar to trypsin. Both these have identical amino acid sequence in the same regions. Chymotrypsin is most active in a slightly alkaline medium. It attacks the peptide bonds which are adjacent to aromatic amino acids such as tyrosine, phenylalanine, tryptophan, and methionine. This results in the formation of short peptide chain fragments. Pancreas secretes chymotrypsin as the inactive proenzyme chymotrypsinogen. It is activated by trypsin in an alkaline medium. At least 4 alternative forms of chymotrypsin molecules are found in vertebrates. They may occur together in the same species. They differ from each other in their secondary and tertiary structure. So, elastase. Elastase is a pancreatic endopeptidase characteristically found in mammals. It is secreted as an inactive zymogen called proelastase, which subsequently loses a small peptide and becomes the active enzyme. Elastase attacks many proteins. Still, it is unique among the pancreatic enzymes in its ability to digest the fibrous protein elastin. Elastin has been detected in chicken and fishes but is found to be absent in cyclostomes. Then carboxypeptidases. These are the pancreatic exopeptidases which attack terminal peptide bonds and remove a terminal amino acid that has a free carboxyl group. They act best on those peptides which contain tyrosine, phenylalanine, or tryptophan. They contain one zinc atom in every molecule. Pancreatic carboxypeptidase are secreted in an inactive form called procarboxypeptidase. They are activated by trypsin or enterokinase. Then aminopeptidases. These are the pancreatic and intestinal exopeptidases which attack terminal peptide bonds and remove a terminal amino acid that has a free amino group. Then dipeptidases and tripeptidases. These are the intestinal exopeptidases which degrade dipeptides and tripeptides respectively, releasing amino acids. These represent the terminal step of protein digestion. Summary of protein digestion in mammals: gastric digestion, protein, gastric pepsin, oligopeptides, casein, gastric renin, curdling of milk, paracasein. Then intestinal digestion: protein, pancreatic endopeptidases, oligopeptidases, protein, pancreatic trypsin, oligopeptides, protein, pancreatic chymotrypsins, oligopeptides, elastin, pancreatic elastase, oligopeptides. Then this oligopeptides, pancreatic and intestinal exopeptidase activity, tripeptides and dipeptides. Later, tripeptides, intestinal tripeptidases, dipeptides and amino acids. Then dipeptides, pancreatic and intestinal exopeptidases turn to amino acids. Then dipeptidases, pancreatic carboxypeptidase create amino acids. Dipeptides, pancreatic and intestinal aminopeptidases create amino acids. So protein digestion in invertebrates. The digestion of proteins in invertebrates, whether extracellular or intracellular, follows the same principle as invertebrates. However, the protein digesting enzyme of invertebrates are not structurally identical to those of vertebrates, although both are functionally similar. A pepsin-like enzyme, which acts in an acidic medium, is absent in most invertebrates, although it has been observed in the maggots of housefly, Musca domestica. Trypsin-like and chymotrypsin-like enzymes have already been detected in several cylindrids, crustaceans, and insect larvae and echinoderms. And similarly, carboxypeptidases and other types of peptidases have also been found in the intestine of invertebrates. So the presence of collagen digesting enzyme, collagenase, is the hepatopancreas of some. The presence of a collagen digesting enzyme, collagenase, is in the hepatopancreas of some crabs is especially significant since such an enzyme is either uncommon in the digestive tract of vertebrates. However, it is present in the venom of several snakes. Collagen digesting enzyme, collagenase.